Hi, I'd like to thank the IWL-CLL committee for giving me this opportunity. My name is Janani Ravikrishnan. I'm actually a graduate student at The Ohio State University. My mentor is Dr. Deepa Sampath, and my project focuses on the biology and targeting of mutant p53 in CLL. So first, a little bit of background. So why is targeting deletion 17p mutant p53 important in CLL? Deletions of chromosome 17p lead to the loss of the tumor suppressor protein p53. This occurs in about 10% of CLL patients at diagnosis. This number increases to about 40% upon progression. Furthermore, 82% of patients that have deletions in 17p also acquire a mutation on their second p53 allele. And lastly, patients that have deletion 17p mutant p53 have poor prognosis, poor overall survival, inferior outcomes in the clinic, and they progress on therapies much earlier than patients that don't have mutations or deletions in p53. So first, we performed exome sequencing on 125 primary CLL patient samples. We see that p53 mutations frequently cluster in the DNA binding domain and are often seen at hotspot sites including R175, R248, and R273H. And these are mutations that are commonly seen across a variety of solid tumors as well as acute myeloid leukemias. Next, we performed a p53 functional assay. So upon induction of DNA damage through radiation, we see that patients that have wild-type p53 have p53 stabilized upon DNA damage and a strong induction of the cell cycle arrest protein p21 after DNA damage. This is in contrast to patients that have mutations in p53, where we see p53 stabilized even without the presence of DNA damage. And we see a much weaker induction of p21 across various different samples. Next, we perform chip sequencing, and we see that mutant p53 samples, our R248Q and our R175H mutants, are recruited to a variety of sites that are critical and important in CLL. We see that mutant p53 samples are recruited to the promoter regions of PKC beta at higher levels compared to wild-type p53. And PKC beta is a really important protein that is found downstream of both BTK and PLC gamma in the B-cell receptor pathway. We also see increase in recruitment to EZH2, a chromatin remodeling protein, as well as BCL-XL, which is an important anti-apoptotic protein. Next, we performed a TAC-seq, where we see that patients that have mutations at R248Q have increased chromatin openness compared to those that have wild-type p53. This indicates that potentially mutant p53 has increased access to the DNA as compared to our wild-type p53 samples. And lastly, we performed RNA sequencing, and we see that there's a differential expression between our patients that have wild-type p53 CLL compared to those that have mutations in p53. Next, we wanted to create a renewable resource in which we could mechanistically study the effects of mutations in p53 without the hindrance of other chromosomal abnormalities. So we use CRISPR-Cas9 editing to knock in both R175H and R248Q mutants in our wild-type p53 CLL cell line called HG3. The Sanger traces here confirm that our edits have been made. And our Western blotting shows that upon creation of these mutations, we see an increase in PKC beta, BCL-XL, as well as EZH2. This indicates that these proteins could be potentially downstream effectors of mutant p53 in this pathway. And we also confirm this using Western blotting in our primary CLL samples, where we also see increases in those proteins as well. So we have PKC beta here, EZH2, and BCL-XL. So lastly, we wanted to use an inhibitor of PKC beta. So MS553 is a potential ATP competitive reversible inhibitor of PKC beta, which Dr. Bird mentioned earlier in this session. We see that treatment for 24 hours with MS553 at 5 micromolar leads to a decrease in a variety of proliferation and survival pathways seen in CLL, most notably a decrease in phospho-PKC beta, phospho-GSK, and phospho-ERK. And therefore, MS553 may be a potential therapeutic option for patients that have mutations in P53. With that, I'd like to say thank you to my mentor, Dr. Deepa Sampath, as well as everyone else in the Sampath Lab. I also like to say thank you to Dr. John Bird and Dr. Jennifer Woyak for all their support in this project, as well as everybody else at Ohio State for their help on this project. Thank you.